Hi everyone, my name is Hakoj. For this section, I'll be talking about spermatogenesis, where I'll be dissecting the essentials and key details involved in spermatogenesis. Spermatogenesis is regarded as the male version of gametogenesis. It encompasses the origin and development of the male reproductive gametes, which involves multiple cellular divisions along with morphological changes. The male reproductive gamete is known as the sperm cell. The sperm cell is unique in that it consists of two morphologically and functionally distinct regions. These regions are the head and the tail. The tail is formed from the cytoplasm of the sperm cell. The tail propels the sperm so that it can fuse with the egg to produce an offspring. The head contains the condensed nucleus that houses the chromosome. Now, depending on the chromosome type, we have the X chromosome and the Y chromosomes. These chromosomes determine the sex of the offspring. This is an image to show the structure of the sperm cell. The head contains an acrosomal cap. The acrosomal cap houses the proteolytic enzyme. This enzyme enables the sperm cell to gain entry into the ovum. The midpiece contains the mitochondria. The acrosomal cap occupies 2/3 of the head size. The size of the head is about 4 to 5 micrometers long and 2 to 3 micrometers wide, while the tail is about 50 micrometers long. The tail contains hair-like filaments that allows propelling of the sperm through undulation movement. This sperm cell is produced in the testes. The major part of the testes include the epididymis, the seminiferous tubules, and the interstitial cells or the Leydig cells. The seminiferous tubules houses what we call the Sertoli cells. These Sertoli cells provide support, blood products, nutrients, and enhances mobility of the sperm cell. The cells forms the starting point for spermatogenesis. The seminiferous tubules are tightly coiled tubules that are lined with stem cells. Now, some of these stem cells form the immature male reproductive cell known as the spermatogonial, while the others retain in the seminiferous tubule to ensure continuity and provision of stem cell. The image right here shows the transverse section of the seminiferous tubule. The spermatogonial that escape the boundaries of the Sertoli cell into the lumen of the seminiferous tubule undergoes mitosis, that is, they duplicate themselves to form the primary spermatocytes. Now, in the case of humans where we have 46 chromosomes, what this means is that the 46 chromosome, which is contained in the primary spermatocyte, is the same as the 46 chromosomes contained in the spermatogonium. What happens next is the maturation of the primary spermatocytes, where they mature, become well developed, and undergoes meiotic division. The first meiotic division produces secondary spermatocytes. Now, they differ from the primary spermatocytes in that the chromosome contained in them is an haploid set of chromosomes, whereas the ones contained in primary spermatocyte is a diploid set of chromosomes. Then, the secondary spermatocytes undergo second meiotic division to form the spermatids. These spermatids then undergoes morphological changes, that is, changes in their shape, changes in their structure to form the spermatozoa. Now, the process by which a spermatogonium undergoes cellular division to form spermatocytes is known as spermatocytogenesis, while the process by which they form spermatids is called spermatidogenesis. The process by which spermatid forms spermatozoa is known as spermiogenesis. Now, we can conclude that spermatogenesis involves 3 fundamental processes, which include spermatocytogenesis, spermatidogenesis, and spermiogenesis. In humans, it takes about 74 days in order to form a mature male reproductive gamete. Now, when these spermatozoa, when they are formed, they are non-motile because they are not well developed. So they are moved from the lumen of the seminiferous tubule to the epididymis. Because they are non-motile, this process is aided by the Sertoli cell. The Sertoli cell is able to secrete testicular fluids as well as peristatic contraction to propel them from where they are being formed to where they can be stored and where they can undergo proper maturation process. The process by which a non-motile matured spermatozoa forms a motile spermatozoa is known as spermiation. Now they become motile when their flagellum is well activated and can propel them through undulatory movements. Now, not all motile matured spermatozoa are able to fertilize an ovum. The process by which a matured motile spermatozoa is able to fertilize is known as capacitation. The key process involved in capacitation is known as alkalinization of the sperm cell. Now, what this means is that the pH of the sperm cell increases, causing the protons contained in it to move out through the ion channels contained in the sperm cell, especially around the flagellum region. This then activates the proteolytic enzymes contained in the acrosomal cap. Now, when they are deposited into the female reproductive tract, tail movement is being suppressed so as to conserve the energy contained in them. But as soon as they get close to the ovum, the tail movement is activated increasingly. Now they are able to know that they are in close proximity because of the presence of a particular substance known as anandamide. This anandamide activates the enzymes contained in the acrosomal cap. Now when these enzymes are activated, they are able to dissolve the thick coat of the ovum. This thick coat is known as zona pellucida. When they dissolve this thick coat, they penetrate, gain entry, and fuses with the ovum. Now what penetrates is only the head of sperm to form the offspring. These are the activities that occurs post-spermatogenesis. On average, about 300 to 400 million sperm cells are being deposited, and the sperm cell is able to stay up to 2 to 3 days within the female reproductive tract. The key details that I've discussed in this section are backed up by these references. It might interest you to want to delve deep.
If you found this video helpful, please give it a thumbs up and subscribe for more evidence-based science content. Let me know in the comments what topics you would like me to cover next. Don't forget, this is the Diary of a Scientist and my name is Akoj. Until next time, keep learning.